All right, well, I want to welcome everybody back. We are going to go ahead and get started with Dr. Emma Kasteel, who is a scientist in the toxicology division at Utrecht University. Emma, take it away.
Thanks a lot. Um, yeah, welcome everyone. I'm very excited to be here, and thanks a lot for the opportunity for me to present our work here. So I will talk today about neurotoxic effects of micro nanoplastics in vitro and some of the past research we have been doing, amongst others, in the Momentum project. I will tell a bit more about those projects as well. Um, Yeah, so I think most people here in the audience and also people watching it later are familiar with micro nanoplastics. So I don't think I have to give a broad introduction about the particles themselves and that there are all possible sources for human exposure and that via these exposures they can get into our bodies and thereby also maybe into our brain. So I think no further introduction is needed here, and I will go straight away to the data. Um, so hopefully that's okay. So for micro nanoplastics in the brain, uh, to get to the brain, of course, they need to be taken up, and yeah, we know more and more that these plastics indeed find their way through our body. So we've seen, of course, some uptake in fish via gills and gut, for example. So you see here fluorescent particles in the gut and gills of fish after exposure. But also we see this in mammals, in liver, kidney, gut, uptake via both lungs and the gut. Um, we think dermal exposure is less relevant, but could also be, of course, a route for the particles to get inside our bodies. So as you see very clearly here that different sizes of particles are ending up in our organs. So then what about the brain? Um, well, approximately 5 to 6 years ago, we did a small in vivo study in mice where we exposed the mice to 1 micrometer and 10 micrometer fluorescent polystyrene particles. And well, we looked at these mice and we looked, amongst others, to the brain. And we found those particles back in the brain already after 1 day of treatment. It was a pretty high dose, but it was oral gavage. So yeah, these particles needed to make their way from the gut to the brain. And they did already after 1 day. And then after 10 days, also, we found them back in the brain for both sizes. So also the relatively bigger particles, the 10 micrometer beads, we did find them back in the brain. But that's in vivo evidence. And then, well, of course, Matt's group came with this study where they detected these particles in human brains. And I'm sure most of you are familiar with this figure, but it was very striking to see that the concentrations of these particles in the brain were much, much higher than in the liver and kidney of the corresponding humans. And when you then look at the different plastics in that brain, you see that there's different polymer types being present. And it's also striking to see that polystyrene is actually just a very small amount of the total polymers in the brain, whereas polystyrene is by far the most researched plastic type. So all the data I've been showing you so far is all on polystyrene because it's the only, or it was the only commercially available particle. So it's the only one you can test if you don't have facilities to produce particles yourself. So research questions we had were, can micro nanoplastics actually cross the blood-brain barrier? They seem to be able to do that because they reach or they make their way into the brain. But there's, of course, also an olfactory route. So via the nose and the olfactory bulb, they can travel into the brain and bypassing the blood-brain barrier by that. So maybe they don't have to cross the blood-brain barrier, but it would be interesting to see if they can do that. We were also wondering if they do get in the brain, do they affect neuronal function there? Because that's, of course, one of the key questions. Is it a problem that there are microplastics in our brain? And the last question we also had was, do aged plastics behave differently than virgin or pristine plastics? So plastics in the environment can age, can weather due to different circumstances like water, like UV radiation, like wind. And we think these plastics may behave differently than just pristine plastics that are very clean. So we did this research within the Momentum project, and Momentum is a large Dutch consortium focused entirely on microplastics and human health. The first Momentum project ran from 2021 to 2024, and we were very lucky to get some follow-up funding. Very grateful for that. So then Momentum 2 started actually while Momentum 1 was still running. So that ran from 2023 to 2025, and the Momentum 3.0 project just started in December last year. And it's a very interdisciplinary project of multiple disciplines in there, and that makes it a very fun project to work on. And so we also had people that could produce particles for us, and we were very happy with that. So we could deviate from the standard polystyrene particles. And we could generate, as we call it, environmentally relevant particles by generating them top down. So starting with a large plastics and milling them down until they get smaller and smaller and into the micro nano size, because that's what's happening in the environment as well. We tested 4 different polymer types: PA, PVC, PP, and PET, in different size ranges. And we also tested the leachates to distinguish any possible effects from particle effects and the chemical effects from the chemicals that are in these plastics. This is a picture of the plastic. So you see the white plastic particles here. This is an example of polyamide, the small particles. And you also see that they have quite rough edges, and they are not the spherical round beads you would see if you would image the commercially available polystyrene beads. So I think these are more relevant for the particles we encounter in the environment as well. So for the first research question, we were looking at the blood-brain barrier. We used an in vitro approach for that, and we used human-induced pluripotent stem cell-derived brain microvascular endothelial cells, or BMECs, and we cultured these on a transwell. So you see here some characterization of those cells. So we stained them for tight junction proteins and some transporters to actually make sure we have blood-brain barrier endothelial cells, BMECs, and we think we actually do. And we culture them on this transwell system. So you have a filter here, and we culture the cells on top. And then we expose them to all the different micro nanoplastics from the Momentum project. So all the polymers I just showed you at a dose of 10 micrograms per ml. And then we look at uptake of the particles in the cells. We look at integrity, and we look at functionality of the blood-brain barrier. So we don't only look if the particles are able to cross the blood-brain barrier, but also if they can harm the blood-brain barrier. Because by doing it, they may facilitate their own uptake, but they may also make the brain more vulnerable, of course, to other chemicals or other particles. So these are some images from the uptake. We did this again with polystyrene because we needed fluorescent particles to be able to visualize the particles, and the Momentum particles are not fluorescent, so we cannot image those. So we used fluorescent polystyrene, 200 nanometers and 1 micrometer. And you very clearly see that both of them are being taken up in the cell. So you also see here the Z-stack. So you see that they're inside the cells in the same focal plane, but not in the nuclei. And as I said, both sides are being taken up. So to look at if the blood-brain barrier is still functioning and still intact, we added a couple of different parameters we measured. First one is the transendothelial electrical resistance, or the TER. So we exposed the blood-brain barrier for 5 days, as you can see over here, the data in vitro. And you see here the TER of the control well. So you see there's a steady increase in TER. It's pretty stable. And then I show you here the data for the different PVC sizes, including the leachates. And what you see is that none of the different sizes actually affects the TER of the blood-brain barrier. We also looked at paracellular permeability. We do that using a quite big fluorescent molecule. And the molecule shouldn't be able to pass the blood-brain barrier. And if it does, it's a sign that, well, your blood-brain barrier is not intact anymore. But you also here very clearly see that none of the PVC particles affects the paracellular permeability. And last but not least, we looked at if the cells were still alive and were still happy. So we looked at mitochondrial activity using an Alderman Blue assay. And also here again, you see that none of the PVC particles affected this metabolic activity. And I can show you all the other graphs of all the other particles. We had more than 20, but I won't bother you with that because they're all very similar to this. So none of the Momentum particles we tested actually showed toxicity on the blood-brain barrier. So that's, well, at least for us humans, I think, good news. that was one slide too far. Then we looked at effects of micro-nanoplastics on neuronal signaling. So as you may or may not know, our brain cells communicate with each other through electrical signals. That's what they do. And if something goes wrong in the brain, you usually see that reflected in this electrical activity. And we have a device called a microelectrode array that can measure that electrical activity. This is how the device looks like. It is quite a simple box, but well, the software and the method behind it is quite complex. So we put a 48 wells plate in this machine. And if we zoom in on that well plate, you see that each well has 16 electrodes embedded on the bottom of that well. We culture our cells on top of this. In this case, these are primary red cortical cells. So cells from the cortex, contains both neurons and astrocytes. You see here the electrodes of the cells actually cultured on top of them. And these cultures become spontaneously active. And we can measure that activity by these electrodes. So it's translated by the software into this false color scale heat map. So you see here the 48 wells in the grid. And each time an electrode detects activity, you see that reflected in this false color scale. And this is then translated to these so-called raster plots. So what you see here is on each row, you see one electrode depicted, so it's 16 rows. And each time an electrode detects a signal, you get such a vertical bar. And we can then derive multiple parameters from this. So we can look at how often a signal is detected, but we can, for example, also look at how synchronized the network is. So you see, for example, over here, I'm not sure if you can actually see my pointer. I'll try to. Yeah, that's better. You see over here that the entire well gives a signal at the same time, and that's what we call a network burst. So then the entire well bursts at the same time. So we can use this assay to look at effects on neuronal signaling, and we did that using two exposure paradigms. The first one is an acute exposure paradigm, and there we first take a baseline recording, and we always first take a baseline recording because, well, each well is its sort of own mini brain, so we always compare within a well. So we take a baseline recording exposed to the micro-nanoplastics in this case, and then measure in the same well again, so we can compare before and after exposure. So in this example, we clearly see that there's much less activity after the exposure. But of course, we are not exposed only acutely to micro-nanoplastics, but for much longer times. And the nice thing about this method is that it's non-invasive, so we can follow the culture for a prolonged time. In this case, 3 weeks is the maximum, because after that the cells are going to die. But we can expose them for 3 weeks to the micro-nanoplastics. So we again take a baseline recording on day in vitro 7, then we expose, and then we measure the activity every 3 to 4 days while they are being exposed to the micro-nanoplastics. So we can really follow the development of the activity over time and compare if that's different, if there are micro-nanoplastics present. And again, we had doses of 10 micrograms per ml for all these experiments. Well, as you can imagine, I have lots of graphs from this because we have many parameters coming from this assay, but I won't show them all to you because I think it's confusing. But to summarize the results is that after acute exposure, we see no effects. And that's maybe not that surprising, because for some particles, we also know that it takes a longer time for the particles to actually reach the cells, so longer than the half an hour we are measuring. But of course, it would be possible to measure effects from chemicals leaching out if that's happening in that half an hour already. After chronic exposure, we did see some effects, but they were very limited. And what I mean by limited effects is that we did see some things that were statistically significant, but we don't consider them biologically relevant. And that's because the effects that we saw were all within the variation of our control. And as toxicologists, we don't think that's then a relevant effect. And statistics tell you that you have false positives sometimes. So if you do enough assays and enough tests, some things will come up as statistically significant, but they are not necessarily relevant. And we think that's what's happening in this case. However, these are just the results of all the pristine particles, and I already told you that we also were interested in aged or weathered particles. So what we did in Momentum 2 is that we took the same polymers and we aged them by exposing them to UV radiation for 1,000 hours. And then they were fractionated again, resulting in the same size ranges. Unfortunately, the smallest size range, there weren't enough particles for us to test, so we couldn't test the smallest size range, but we could test 1 to 5 and 5 to 10, and also again the leachates. So we tested those, and now I will show you some graph from the microelectrode array. But I first want to explain to you how to read such a graph because they can be a bit complicated. On the x-axis, you will see the time, so the day in vitro. And then on the y-axis, you will see the percentage of control normal development curve. So in here, the black line is the normal development of a culture over time, so it's the control, and the gray shaded area shows you the variation of that control. And everything that's above that control line is what we call hyperexcitation, so there's more activity, and everything that's below that control line is what we call an inhibition. And this is an example of the limited effects we saw for the pristine or the virgin particles. So this is PET. So you see here that the leachates and the 5 to 10 micrometer particles showed a statistically significant increase in activity at the 2 latest time points. But you also see that it's still in this gray shaded area, so that's within the variation of our control. So that's why we don't think this is a biologically relevant effect. Then if we compare that to the UV aged, it's actually striking because we see the same particle size popping up. So the UV aged PET 5 to 10 micrometer, same as for the virgin. We also see an increase, and it's statistically significant for the 2 latest time points, but it is again within the variation of our control. So also here we don't really think this is a very biologically relevant effect. For the other UVH particles, we see similar things. So we do see some things happening here and there, but it's all happening within this gray shaded area. So we don't see any very shocking inhibitions or hyperexcitations. So to conclude, I hope I'm doing okay on time. Good. What I've showed you is that we see that polystyrene micro nanoplastics can be taken up by the Bmax, so by the blood-brain barrier cells. But none of the tested momentum micro nanoplastics affect blood-brain barrier functionality or integrity. So the blood-brain barrier remains intact and functions. We see no effect on neuron activity after acute exposure, both for pristine and aged particles. And we see very limited effects on neuron activity after chronic exposure. But the relevance of those effects is still a bit unclear to us, and for now we don't think they are very relevant. I do have some considerations, though, with all this data. One question you might have is how about translocation of the blood-brain barrier? Why didn't you look at that? Well, there are a couple of mostly practical reasons, actually, that we didn't do that yet. It is definitely still on the wish list. One of them is that we need fluorescent micro nanoplastics for that. We need to be able to visualize the particles. And so for now, that's only possible with the polystyrene. And we are also limited by the pore size of the transwell. So I showed you the schematic with the transwell system, and we have a filter, and we grow our cells on top of that filter. But that filter has an average pore size of 400 nanometers. And that means that particles that are bigger than that will get stuck on that filter. And you will never be able to measure translocation of those particles into the basolateral compartment because you just cannot get through those pores. And those are all very practical limitations that we were never ever encountered before with when doing research with chemicals. But now with the particles, these are things we run into. But I think that's very common to the microplastics field to have these practical issues. Another consideration, and I think it's also known for all the people working in this field, is that it's very complex to do this research, because the fact that we don't see effects now doesn't mean that none of the micro nanoplastics that exist affect the brain or the blood-brain barrier, because we tested quite a few different polymer types and sizes, but we are nowhere close, of course, testing all the different materials and all the different uh, micro nanoplastics that exist with all their shapes and colors and sizes and additives and etc. And dosimetry, of course, also is complicated in this field, and in vitro dosimetry is an important thing to consider when doing this research. For example, we tested polypropylene, but polypropylene has a density smaller than 1, so it's buoyant. So you have to think about that, because otherwise the particles will never be in contact with your cells if you have a 2D cell culture and your cells embedded on the bottom of that culture well. So those are all things that are important to consider and you have to take into account while doing this research. But as I said, we are very lucky to get follow-up funding for Momentum. So we just started Momentum 3.0 and we will address at least some of these issues. So we will test much smaller plastics, so really nano-sized, mostly polyamide, because we saw in Momentum 1 and 2 that polyamide is actually seems to be quite toxic to the lungs. So we wanted to do more research into that and also focus more on the brain to see what's actually happening there with these very small particles. And I wanted to conclude with some things about literature. We recently published a literature review on the neurotoxic effects of micro nanoplastics. And this is a table from that review where we tried to summarize the effects we see. So we just focused on rodent, in vivo, and in vitro studies. In vitro, all human and rodent based, and in vivo, all rodent. And this is just a summary of the results we saw. So the color indicates how many studies saw significant effects, and the number indicates the number of studies that actually looked at that. And what you then see is that most studies look at very general toxicity pathways like cytotoxicity, oxidative stress, neurotransmitter signaling. But what we also saw is that polystyrene is very dominant in the studies. I already briefly mentioned it, but more than 85% from the in vivo studies and more than 90% even for the in vitro studies focused on polystyrene. And well, if you remember the first graph I showed you with the data from Matt's lab, is that polystyrene is definitely not the most relevant particle. I think not for human exposure in general, but also not for the brain. So it's important to start focusing on other polymer types as well, I think. What we also saw is that almost all these studies just use pristine particles, and this is also not what we are exposed to in the environment. So I think it's also important to start focusing more on aged particles. Dosing levels, I put a question mark there because it's difficult. We don't have fully mapped what human exposure looks like, I think. The dosing levels in most of these studies is super, super high, and therefore probably not really comparable to the actual human exposure levels. And quality of studies is also, I think, quite a big issue. We encountered 829 studies in this for this review, but we excluded 738 of them, and they were excluded based on quite basic quality criteria, like mentioning the polymer type or mentioning the size, or very basic things that are really, really basics you at least need to mention while publishing research on micro nanoplastics. So those are some things I wanted also to mention here to make sure to be critical of the literature you encounter while studying the micro nanoplastics. So that was actually all I wanted to tell. I really would like to thank the people in our group, the Neurotoxicology Research Group. You see most of them on this nice picture over here. And I also want to thank the Momentum Consortium, which is still a great consortium, still in Momentum 3. And of course, SunMW for funding those projects. And that was all I wanted to tell. I hope it was a clear presentation, and I'm very happy to take any questions.
All right. Fantastic job, Emma.
Thank you.
There's about 5 minutes and a few questions in the chat. I'll read them. What I'll ask is if you can give us relatively quick answers, we can maybe get to all of them.
Yeah.
But Aura Newman was asking if you have looked at or observed any protein or neuronal aggregates in your assays.
We haven't looked at that yet. We are planning to also look at some protein aggregation and amyloid beta related things, also in relation to neurodegeneration and Alzheimer's disease. But we haven't looked at it yet.
Cool. Do Young Kim is asking, is it possible that the lack of glial cells, astrocytes, microglia in your system could contribute to the negative results that you're seeing?
Yeah, that's a very good question, and that could definitely be the case. So there's quite some astrocytes in our system, but not a lot of microglia. We did some separate tests with microglia to look at cytokine secretion, and there we didn't see any increase in cytokine secretion, but we used a not ideal microglia cell line. So it's also somewhere on our wish list to do more extensive microglia studies in relation to micro-nanoplastics, because I think the immune system is very interesting in this field, and therefore also the immune system of the brain. So yeah, super interesting question, and definitely something we would like to look at.
Okay, cool. Do Further is asking, can the increase in the network burst activity in your MEA measurements be the hyperexcitability? Have you done any follow-ups on the time course of individual electrode activities?
Um, yes, partly. So we did dive into the data, and we have around 40 parameters coming out of this data besides the network burst. But for now, the data is not really showing anything to us that there's actually anything happening. So we still think that what we see is sort of arbitrary thing. But we want to, before publishing this data, dive into it even more and see if we can derive some patterns there to say something about if there's actually something happening or not.
Okay, great. Sammy Romanek is asking if can you tell the size of the dye you used in the permeability assays? Were you using a dye or just the electrical resistance?
We used both. So no,
Okay.
we used the electrical resistance, and we used sodium fluorescein
Mhm.
as a paracellular permeability assay. So I don't know by heart what the size of sodium fluorescein is, but I think you could Google that.
I seem to recall it being around 400 Daltons, but
Yeah,
yes,
I think it's something like that. Yeah.
and she's also wondering, are you able to perform longer studies beyond 3 weeks in your assay?
Currently, no, because the primary cultures we are using just at one point don't really survive anymore and don't show activity anymore. But we are currently transferring to iPSC-derived cortical cultures for the microelectrode array as well. And there, then we could definitely try to prolong our exposures.
Okay, very cool. And I have one last question, which we've talked about before, I think, which is you're growing your cells in the MEA dish and then you're treating them with the plastics, but can you grow them in the presence of the micro and nanoplastics and then look at function?
For the microelectrode array, you mean?
Yeah.
Yeah. Yeah, with the iPSCs, that's definitely possible. So for the primary cultures, it's a bit harder because, well, they are coming from the brain of the red pups, and then so they aren't really growing anymore. We just plate them, and then that's it. They need some time to acclimatize, but that's it. But for the iPSCs, we could definitely try to start exposing them from the beginning on. And then even when there's no activity yet, so we cannot measure any activity on the MEA, we can still expose them and then see if activity develops differently. Yeah, that would be very interesting.
Very interesting indeed. All right. Well, thanks so much, Emma.
Yeah, you're welcome.
All right.